Hi, my name's Luna Smith, and I'm going to be presenting on the BRCA gene and making some AP biology connections. And my partner for this project was Maya Paskey. So here's some photos of us skiing together and us at prom. So the first unit we learned about this year was the Chemistry of Life unit. And this unit taught that macromolecules make up all life on Earth. And these four macromolecules are carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates make up simple sugars, lipids make up fatty acids, proteins make up a lot of the enzymes in your body, and nucleic acids make up your DNA. And so the proteins in our body are determined by our DNA, and they're made up of amino acids. And once again, our DNA is made up of nucleic acids, and it's the order of thymine, adenine, guanine, and cytosine, that the nitrogen bases in our DNA, and it's unique in every single person. So in these proteins, whose recipe is in DNA, there's primary, secondary, tertiary, and quaternary structure levels. So primary structure is just your string of amino acids. There's no folding of any kind. It's just your entire string of amino acids. Secondary structure is when your sequence of amino acids starts to fold up because of hydrogen bonds between amino acids, which is the bonding of hydrogen and oxygens. It's a weaker bond. It's between hydrophilic molecules. Tertiary structure is when your protein starts to take on a lot more folding. This is when you have hydrophobic interactions, disulfide bridges. You're having a lot more folding. And also, again, for secondary structure, you can have a beta pleated sheet or an alpha helix to add on to that. And quaternary structure involves multiple strings of amino acids. So this involves like multiple tertiary structure, like not full proteins yet, coming together to form a fully functional protein. And as you may know, protein form affects function. So anything from pH, salinity, or temperature, which changes the form of a protein, affects how it's going to function in your body. And to connect to the BRCA1 protein, which is functional in most people, but it's often dysfunctional and can be linked to a higher risk of breast cancer when it's dysfunctional. This protein has a quaternary structure, so it's made up of multiple strings of amino acids that come together. Next up is Unit 2, which is cell structure and function. And so the cell membrane, the fluid mosaic model, includes a phospholipid bilayer, which use negatively charged phosphate heads and fatty acid tails, which are lipids. It includes proteins. There's a bunch of different types of proteins that help with transport. There's a bunch of proteins on the cell membrane. And next is cholesterol, which helps maintain like the fluidity or solidity or solidness of the plasma membrane. And we often think like, don't eat cholesterol, it's bad for you, but in fact it's very important in keeping your cell membrane at the right temperature. And so molecules can travel in and out of the cell through this phospholipid bilayer. And so if you have proteins for export or vital molecules that need to come in through the cell, they're going to come through the phospholipid bilayer, whether that be through simple diffusion, so non-charged molecules like oxygen or carbon dioxide, or if it's going to be a charged molecule like water, you'll go through an aquaporin protein. Or if you're a larger molecule like glucose, you'll go through a transport diffusion or a facility diffusion through a transport protein. And so once you get inside of the cell, there's a bunch of different organelles that work together to carry out cellular jobs, which help keep the cell running, creating the nutrients it needs, bringing in nutrients it needs. It's very important that all the organelles are working together. And so different parts of the cell are compartmentalized to allow for cell efficiency. So you have the endomembrane system, which involves the making of and exporting of proteins. You have waste organelles like the lysosomes. You have energy organelles that help create energy in the cell, like the mitochondria. And so when we talk about the BRCA1 gene, the BRCA1 protein is found in both the nucleus and the endoplasmic reticulum. It was used to be thought that it was only in the nucleus to help repair DNA damage, but it's now found that the functional protein also works at the endoplasmic reticulum, which sorts and which helps it's a place of protein production for ribosomes on the rough ER, and the smooth ER helps detoxify the body. So next up is Unit 3, which is cellular energetics. So in the body, there are enzymes, which are proteins, and they help increase reaction rates inside of the body without being absorbed by the reaction rates themselves. So substrates will enter into an enzyme, and then they will come out as products. And so when we talk about metabolism, we're talking about all the chemical reactions that happen in the body, whether that's catabolic, which is energy releasing, or anabolic, which is energy taking. And so one of our catabolic reactions is cellular respiration, which helps create ATP for the body. And so cellular respiration kind of has 4 main stages. There's glycolysis, which is in the cytoplasm of the cell, and it breaks down glucose into pyruvate. Then we have pyruvate oxidation, or the link reaction, as kind of the pyruvate is kind of like entering into the mitochondria, some CO2 comes off. Then we have the Krebs cycle, which just basically takes a lot of CO2 off of the pyruvate to reduce NAD plus to create NADH and FADH, which will go to the electron transport chain and provide the electrons that allow for the flow of electrons to help pump hydrogen into the intermembrane space of the mitochondria in order to hydrogens to flow down ATP synthase back into the matrix so that ATP can be produced for the body. And so, again, this is a catabolic reaction, and it makes ATP, which you need for energy in your body. And so when we talk about the BRCA1 gene, it's found that the mutated BRCA1 gene has also found to impair mitochondrial function, which would create less ATP for the body, less energy for the body, which can be seen in some of the cancer symptoms that people may be feeling, fatigue, because they have less ATP available. Next, I'm going to talk about Unit 4, which is the cell cycle and cell communication. So cells progress through G1, S phase, G2, and mitosis during the cell cycle. G1 is when cells are growing. They kind of have just gotten out of mitosis. They're growing. They're getting bigger. In S phase, all their DNA is replicated, so that they're kind of preparing for mitosis. They have that replicated DNA for when they're going to divide. G2 is when cells kind of get their organelles prepped. They're prepping for mitosis. They have certain organelles that are needed for mitosis are prepped. And then mitosis is obviously when the cell separates. You have prophase, metaphase, anaphase, telophase, and then cytokinesis. And so when we talk about cancer cells, with people who may have the mutated BRCA gene, cancer cells progress through the cell cycle uncontrollably, meaning they don't stop at any of the checkpoints to check if their DNA has been properly replicated. A lot of times there's a checkpoint right after the S phase to make sure that all of the DNA in the cell has been properly replicated, and if it hasn't, then the cell will either repair the DNA or go into apoptosis with the P53. But a lot of times cancer cells just progress through the cell cycle uncontrollably. They're continually doing mitosis. More and more cells are going through, and the DNA is damaged. It's mutated, so that's not good. And cancer cells also don't follow anchorage dependence or density dependent inhibition, and this basically means that cancer cells they can't feel the cells around them through cell communication, and they don't have an idea of density, meaning they build up, and they can't really tell when they're touching other cells, so they're just kind of expanding, and they don't have like a awareness about what's around them. And so the normal BRCA1 gene creates a protein that acts as a tumor suppressor, which again is going to be making sure that the cell is not replicating even when the DNA is damaged or mutated. But the mutated BRCA gene can't repair DNA damage, so it allows the cell to keep on progressing through the cell cycle with DNA that is damaged, which is not good. So next, I'm going to be talking about bioethics. So why people would opt out of genetic testing is that testing positive for the BRCA allele can lead to genetic discrimination, meaning in the workplace or at your employer, if they know that you have a mutation, if you have the BRCA allele, you're more likely for breast cancer, meaning you may not be able to work there as longer, you may have to be spending time away from work as treatment. So it could be dangerous to test positive for the BRCA allele, even though you don't even know if you're going to get breast cancer. Testing is also expensive. It's not financially covered all the time by insurance, so a lot of families may not want to spend money on something that may not even determine if they get breast cancer or not. And going off of that, only 10% of breast cancers are due to BRCA, meaning there's so many other environmental factors, other genetic factors that could be causing you to gain breast cancer. So testing positive for the BRCA allele isn't like you're going to get breast cancer. It's more of a risk factor that you have when you have to be considering how you're living your life. And so if you do choose to test for the BRCA allele, some positives is that if the allele is present, you can take preventative measures, you can get treatment early, you can realize how maybe environmental factors are affecting you, and kind of take preventative measures to maybe slow the possibility of getting breast cancer or lower that risk. And if you know you have the BRCA allele, you can kind of know how that's going to affect your offspring. If your partner has a BRCA allele, you can see if your child is going to be heterozygous dominant for that, or if they're going to have the allele, which it could influence like who having children and how that affects how your genetic condition will affect your children. And so my picture here is George W. Bush signing the GINA, which is the Genetic Information Non-Discrimination Act in 2008, which actually made it illegal to genetically discriminate people, which is very pertinent to genetic testing and how getting a positive genetic test for something like the BRCA allele will affect even your income. So yeah, thank you.
Now we're going to talk about Unit 5, which is hereditary. This unit was generally about the modes of inheritance, which are autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive, and identifying whether the trait is one of those. And in this lab, the method of inheritance for breast cancer is autosomal dominant. Another is alleles separate during meiosis II. So they separate in meiosis II in order for each gamete to acquire one of the two alleles. Another is law of segregation and law of assortment in meiosis. So law of segregation is the two alleles for each gene separate, while the law of independent assortment is the alleles of the genes on non-homologous chromosomes assort independently. My team is focusing on how it only takes one copy of a mutated gene to develop breast cancer. So if any allele on a gene has a mutation, it only takes one copy of that gene to have a person develop breast cancer and to pass it on. But only 10% of breast cancers are hereditary, so that's a good thing. And we're also focusing on pedigrees, which are analyses that can identify patterns of inheritance, how from the knowledge we have received from the family's older generations affect the newer generations to see who potentially is a carrier has breast cancer. So connects to the photo on the right, as you can see, here is a pedigree of a family who has breast cancer, and we are able to see from knowledge that people in the lab are telling us to see who might potentially have it. In the lab, we used the mom's knowledge of who had breast cancer and who did not in her family in past generations and now generations. And we used a pedigree chart to help determine who potentially has breast cancer based on the pattern of inheritance and which is autosomal dominant, and make an assumption on who in the family has breast cancer before we went to gel electrophoresis. Okay, next is Unit 6, Gene Expression and Regulation. This unit in general is about how gene expression involves the process of transcription, which is DNA transcribed to form RNA, and translation, where RNA is translated to form proteins, and how the information from a gene is used to synthesize proteins. Okay, so cells regulate which genes are turned off and on in response to internal and external cues. So there are different types of regulation within cells. There's transcriptional regulation, post-transcriptional regulation, translational regulation, and post-translational regulation. So each part of the gene expression process is regulated. Another is mutations. And mutations are changes in DNA sequence that can affect gene expression and the resulting proteins. So on the right, there is a picture of a cell with multiple DNA mutations. In that, there could be a silent mutation, which is the mutation that does not change the amino acid sequence of the protein, a missense mutation, where it's one change in the amino acid protein, which affects their protein function. Nonsense mutations can lead to non-functional proteins. There can be a frame shift mutation, which is an insertion or deletion, and then it alters the entire amino acid sequence downstream. There can be duplications, where a segment of DNA is duplicated. There can be a deletion, where a large segment of the DNA is deleted, and inversions. A segment of the DNA is reversed within the chromosome, and there can be translocations. A segment of the DNA is moved from one chromosome to another. So there are many different types of mutations, and any one of these can lead to the formation of tumors. So my team is focusing on how the errors in DNA replication cause cancer and how these mutations can lead to the formation of tumors and how the overexpression of genes drive growth of breast cancer. So when genes are overexpressed and not regulated, it drives growth of breast cancer, non-stop breast cancer. So in the lab, we use gel electrophoresis to identify mutations in each person's gene and identify how separated the DNA molecules are, and to identify the difference in fragment size to spot mutations as well. So now is Unit 7, which is Natural Selection and Evolution. General knowledge about this unit is that favorable traits increase survival and reproduction. There's genetic variations, where mutations in sexual reproduction create diversity. There's comparative anatomy, where structures indicate common ancestry. There's also adaptive evolution. Selection improves organism environment fit. And my team is focusing on epigenetic changes, where it's subject to natural selection and is heritable across mitosis and can affect the fitness of cells by changing its phenotype. Adaptive evolutions, therapeutic resistance. So as you can see on the right, where then there's natural selection of tumor variants, and then where the tumor isn't growing as fast, and then the second, and B is when natural selection is immune. The tumor is immune to like therapies like radiation therapy, and it just causes genomic instability, and then the tumor spreads. And then high genetic diversity within neoplasms predict both faster progression of reduced patient survival time. So the higher the genetic diversity in neoplasms, the faster progression of reduced patient survival time is. So in the lab, we are looking for evidence of mutations driven by natural selection over generation. So through electrophoresis, we are able to see what maybe if the grandfather went through chemotherapy, and then if the tumor is another variant of the breast cancer tumor because it evolved and became resistant to the therapy, to see what potentially the breast cancer tumor would look like for all of the daughters and the mother. Okay, and Unit 8, which is ecology. So just general about this unit is that there are community interactions, predation, competition, symbiosis. There's mutualism, communalism, and parasitism. Mutualism is where neither of them benefit or unbenefit. Communalism is one benefits and where they both benefit, and parasitism is where one benefits and another doesn't. There are human disturbances, which is over-harvesting invasive species and global change. People over-harvest and lead to the ecologies and the niches changing dramatically. Invasive species, people bring invasive species to different places and they invade and reproduce really quickly and change the niche as well. And then there are global changes like pollution and greenhouse gases. There's also responses to environment, the types of behaviors in animals, and the responses in plants. So we're focusing on how the exposure of pollutants, which is a human disturbance, or chemicals in the environment, cause mutations in the BRCA1 and the BRCA2 genes. So the exposure to pollution or chemicals can lead to mutations in the BRCA1 and BRCA2 genes, which then cause it to be repressed and then a tumor to spread uncontrollably. And the interactions between cancer cells and the surrounding tissues. So interactions with the immune system, this is definitely parasitism because the cancer cells are spreading uncontrollably and the immune system is shutting down. And then how in connection to the lab, the expression of biomarkers there and how the environmental factors influence the expression of the biomarkers. So for each person's DNA, we don't know what they have been exposed to, if they have been exposed to pollutants or chemicals or exposed to something else. So trying to see how environmental factors influence the expression of the biomarkers and if that changes the results of the gel electrophoresis. So the picture on the right is breast cancer risk factors, and as you can see, there's definitely genetics, which is shown in the lab, and radiation, which is a really big thing. Exposed to radiation can cause mutations and cause mutations in genetics, which can then be passed down to your kids. For my bioethics slide, I talked about the bioethical concern. So the problem of overdiagnosis and overtreatment, the problem of underdiagnosis and undertreatment, and the results of false negatives and false positives is a huge thing when doing genetic testing. There's two perspectives for this. One is opting out of genetic testing. There's discrimination within insurance companies. There's discrimination between your work and family and anything when you are diagnosed with a certain disease. Finding out about the allele gene has profound impact on a person and their family. If you have it, your family doesn't know who needs to get tested, and it could cause chaos in families. And especially if it was a false positive, then that causes a lot of chaos as well. It also might have consequences on daily life, like your work, your boss. You could cause discrimination in your work. Families may fall apart and cause chaos, and it may cause feelings of depression between families as well. And then in your marriages, if your other partner may not want to stay with you, if they think they have to deal with that. And so that's definitely these are big bioethical concerns with genetic testing. But another perspective is choosing to do genetic testing because it can catch possibility of future diseases, not just the disease that you may have, catch possibility of future diseases. So it can also help catch cancer or any diseases early to help fight it off early instead of dealing with it later. And then you can know if your children inherit it. If you do genetic testing and you find out you have this disease, and you can get your children tested as well to see if they have it, to start taking precautions in the early steps to either not contract it or develop it, or to help limit it and fight it off early.